Welcome to SARC Talk. SARC is a nonprofit cancer research organization that develops and manages clinical research trials in pediatric and adult sarcomas. SARC Talk is a podcast to discuss activities of SARC, interview sarcoma research leaders, and review scientific initiatives in the sarcoma field.
Welcome to SARC Talk, episode number 20. My name is Scott Okuno, a medical oncologist at Mayo Clinic and Chief Medical Officer for SARC. At SARC Talk, we interview interesting people who are connected to sarcoma, where we can all share and spread the knowledge about sarcoma. I'm pleased today to have Dr. Candace Haddox, a medical oncologist at Harvard Medical School and at the Dana Farber Cancer Institute, with us today. Dr. Haddox graduated from medical school at the University of South Florida, where she was first exposed to sarcoma research with Dr. Damon Reed at Moffitt Cancer Center. Damon is now at Memorial Sloan Kettering. She did her internal medicine training at Mayo Clinic Rochester in Minnesota, and she went to Duke to complete her fellowship in oncology and was chief fellow there. She then came to Dana Farber in 2021 as a member of the Division of Sarcoma and the Phase 1 program, and the Center for what they would say is Cancer Translational Innovation. She's a clinical and translational researcher developing clinical trials and has a passion for teaching and mentoring. Please welcome Dr. Haddox.
Thank you so much. It's great to be here, Dr. Okuno.
Yeah, well, thank you very much. And, with some technical hurdles, we're all here. So this is all that matters to us. So, Dr. Haddox, our paths did cross back when you were here at Mayo Clinic Rochester as an internal medicine at Mayo Clinic Rochester. And you worked with us on a paper called Exoskeletal Osteogenic Sarcoma and did a review. And I think Dr. Robinson and you worked quite heavily together on that. So that's when we started to know about you a little bit more. In fact, I was also at ASCO this year, of all places. I met my colleague, Dr. Matt Getz, who is a medical oncologist in Rochester, who also said he remembers you as well. So there's a lot of connections, and it's funny that I had to go to Chicago for Dr. Getz, my colleague in Rochester, to tell me about you as well. So we're really excited about that. And maybe, as we're having a discussion here about looking back at how you got here, and I know part of the discussion is that you actually started, above all things, when you were at medical school. Maybe you can share a little bit of how that happened and how Damon kind of shaped your career at that point. And then maybe a little bit more what you learned in internal medicine here at Mayo Clinic Rochester.
Yeah, absolutely. So I was in medical school at University of South Florida, as you mentioned, and one of the philosophies of our medical school was to integrate learners into medical practice very early. So just by chance, I was paired with Dr. Ricardo Gonzalez, who is a sarcoma surgeon, and probably within the first couple of weeks as a medical student, those were my earliest exposures to medicine, was doing these really large sarcoma operations in the retroperitoneum. So that's where I first learned of sarcomas and got connected with Dr. Gonzalez, and then he connected me with Dr. Reed, and he was a wonderful mentor, had a project in mind that I could help get involved with, and that was the osteosarcoma study, looking at differences in outcomes between pediatric patients and young adults. And from there, it's just, I feel like I've followed the sarcoma community and vice versa, and now here I am today. But that was my earliest start. Then when I moved to residency at Mayo Clinic in Minnesota, stayed in touch with Dr. Robinson, who was actually one of the individuals who interviewed me for residency. And we worked on some projects together. I rotated in his clinic. I interacted with so many wonderful people at Mayo that really shaped me both clinically and as a researcher. And I think a lot of what I learned at Mayo was really how to take good care of patients. I really felt that was the core mission at Mayo. And I think that I came out of internal medicine training with a lot of those values. We had a lot of very complicated medical issues that we were managing, collaborating with multiple specialties, thinking outside the box. So these were all like key foundational things to learn as a resident, that I feel like I've carried with me, and I'm so grateful for. As you know, the environment at Mayo is so collegial, so it's been wonderful to work with so many colleagues there and connect with them over time. I think I ran into Dr. Getz as well at ASCO. It was so great to catch up.
Yeah. Well, maybe you can share what award you did receive while you were at ASCO.
Yeah, I got one of the career development awards there for a project that I'm working on, and just specifically SARC 44, which I know we'll talk about a bit more.
Yeah. So it's an exciting time when a lot of us are in a community. We all kind of know each other as we're going through, and it's just great to celebrate, that success that you, with a lot of other members, help support for you with at the ASCO Award. So that was really exciting. Matt was so proud. So just to let you know, it was great.
Thank you.
You then, I agree. It's good to hear that. I also trained in internal medicine at Mayo, and the values that you shared about how to take care of people, how to respect each other, kind of plays out in your career. You then went over to Carolinas there and went to Duke. And as a fellow there, you learned oncology, but you're also a chief. So maybe you can share with the group how that was and what you learned about yourself or what you learned about your colleagues as a chief.
Yeah, absolutely. I came to Duke in 2018. Our first year was very clinically heavy. We learned a lot about oncology, rotated through all the different disease groups, and just got a broad foundation in oncology. And then the last 2 years of fellowship were meant to be for research. But as you know, it was a unique time, not just in my training and career development, but for the world. We were going through a pandemic. So that changed things. And, I think while my initial outlook was to get into the lab, to start a lab-based research project and grow my career in that direction, unfortunately, with the pandemic, things were shut down. We had to make a lot of changes, so we had to be adaptive. During that time, there was some discussion, not because of the pandemic, but just in general, of having chief fellows. And once the pandemic hit and we had to be adaptive, like I mentioned, it was then a really essential role. And I had the opportunity to begin as a chief fellow at Duke. And we had to sort of restructure certain things, try to keep our clinical mission moving forward, but also maintain the education, the research and career development for the fellows. So it was a time. I'll mention Dr. Rich Riedel was one of my mentors at the time, clinically and also through all of this. He was also new to the role of fellowship director, and had to quickly adapt to all the changes that were going on. So we really formed a good team, and we're very collaborative and thoughtful, and I'm so grateful for that time, because I came away learning a lot, not just on the research front and what my interests were, and continuing to learn more about sarcomas, but also how to keep certain aspects of the institution moving forward, and being involved in institutional service, which is an important part of academic medicine too. So fellowship was a great learning experience for me, for sure.
Well, we're very happy about that. And thinking back, your career in medical school, internal medicine, and hematology and oncology, you were mentored by sarcoma doctors. Damon Reed, Dr. Robinson, Dr. Riedel. So you've had that exposure all the way through, which is great. And that was so great, you decided, hey, I want to go to a different institution. So maybe you can share kind of some of the aspects or the factors that went into going to Dana Farber at the Harvard system. And yes, they wanted you, but let's just assume that they wanted you. How did you decide that you decided to go to basically a different institution?
Yes, that was a tough decision. And what I was looking for when I was considering the next steps in my career was really looking for an opportunity to have strong research resources and support to achieve my academic goals. And I think I could identify those features of many of the opportunities that I was considering. But when I was getting to know the program here and the individuals here and knowing the legacy of the group and the collaborations internationally and the opportunities to partner with lab collaborators here, that was something that stood out to me that I really wanted to, have early on during these formative years of my career. Another unique opportunity that I have at Dana Farber is I'm the sarcoma representative in the Center for Cancer Therapeutic Innovation, which is our phase 1 group. So our phase 1 group is structured, and I think a little bit of a unique way compared to other programs. I see patients with sarcomas only and put them on phase 1 studies, but have access to this larger portfolio where the drugs may ultimately be under development for breast cancer. But as a sarcoma specialist, if I can envision a role for a patient with sarcoma based on sarcoma biology or genetic factors or biomarkers, then I have the opportunity to put patients on those studies early. And that's a unique role that I play in our group here as I'm the sarcoma representative, and it's been really exciting to be part of early drug development. So that's how I ended up here. I'm very fortunate to be part of this group.
Well, we're very happy that you're there and thriving and enjoying yourself. You did say that earlier on that there was some inkling that you might want to do bench research. And, things happen, COVID, et cetera, change. But with your involvement with what I would say the phase 1 studies and drug development, how sad are you, or not sad, that you didn't pursue a career, and it's never too late, but a bench lab part? Because you said you came to Dana Farber with this idea that, hey, I have collaborators that can do that work, and then my skill set is maybe different. So can you share?
I think that my background in all the lab research that I've done over the years has given me a strong foundation in the language, the techniques, how to think like a scientist, how to ask questions, how to form hypotheses and answer questions. And I can now leverage that into putting it into practice. And I hadn't explored this until I joined our group here. So I didn't know what that would feel like, but I really enjoy it. So I think it's a great fit for me. And I do, like I said, get to collaborate even with SARC 44. Dr. Jonathan Fletcher is a key scientific collaborator on this study. And it's been so rewarding to work with him and his group. And we can talk science, we can talk signaling, we can talk all the things that I find really exciting and interesting on the scientific front. But I feel like it's so cool to sit and talk with a patient, to tell them the rationale, to have this insight into the scientific lab and what happened there and what formed the basis of putting the study together, and then to put them on the trial and see how they do. I think that's such an exciting part of this trajectory to be on. So I'm very happy with how things turned out. I think I'll always miss holding a pipette and designing experiments and doing all those things. I have such an appreciation for that, and I do miss it for sure. But I think a lot of the bigger themes of being involved in the questions and finding the answers in that pursuit is satisfied by doing the phase 1 and translational work that I do, that it's a good fit.
Yeah. For those that are just listening, you can see Dr. Haddox's face shine when she starts to talk about the opportunities in the phase 1 and the mechanism, the signaling that, really helps drive science, and we'll get to that. We always say that the sarcoma community is very small. However, it's big, okay? And it's sometimes hard to navigate within your institution where you are with sarcoma, yet alone navigating outside of your institution. And being an expert in sarcoma, there's only 100 different sarcomas that you have to be proficient at, and at Dana Farber, you probably see 130 different types. But anyway, there's a lot of learning just to get up to speed there, and you have a great group to help support that. We're trying to explore ways that we can connect in the sarcoma community. And how did you navigate connecting within Dana Farber? You kind of suggested ways that you've done at other institutions, but how have you started to connect with other groups outside of Dana Farber? Because I know you're involved with other groups as well.
Yes, I have to point to my mentors both within Dana Farber and outside of Dana Farber for making those initial connections. I felt like early on I had pick of the litter for what opportunities I wanted, and there were so many. And my mentors here and others in the disease group have been so supportive and saying, hey, why don't you look into being part of Alliance, come to a few meetings, see what SARC is all about, hear a little bit more about ETCTN trials and what CTEP offers and those resources. So I was, invited from my mentors to come join, see what felt like a good fit, see where my interests were. So it was, I have to thank my mentors for that, really. And then some other opportunities have come around. Early on, I developed an interest in desmoid tumors also. I think that's a tumor that's very fascinating, how they can, grow or regress spontaneously, and was really interested in that. So started expressing these interests, and then had the opportunity to join the Desmoid Tumor Research Foundation Medical Advisory Board, which has been very rewarding. Being involved in their annual meetings. They're such a enthusiastic group. So I think it's a little bit of, having great mentors, but also not being shy about just seeing what's out there and what fits. And as you know, the sarcoma community is so welcoming and collegial and supportive, and it's been really easy to get integrated and branch out. So
No, that's exactly correct. And a couple doors are opened. You're able to walk through, not missing a beat. You're in there. You're doing the things you need to do and have done, so which is absolute. And part of what SARC is trying to do too is some of our young investigators, young sarcoma doctors, might not have had the mentors to open those doors because they're starting brand new programs up. So we'll continue to work with them and hopefully provide mentorship to open the doors. But that then kind of gets to the point where you have been a mentor as well, because you've learned some skills. You had the skills probably. You were honing them in by being mentored. And maybe can you share how you've been giving back now some of the stuff, and how has that shaped how you develop a mentee relationship rather than a mentor relationship, or vice versa?
Right. Yes. Yeah. I'm starting to make that transition a little bit, and have a few fellows who are interested in sarcoma that I've been working with. And I'm trying to be really thoughtful in articulating the things that my mentors did successfully along the way, and trying to apply that to how I'm interacting with mentees. I think one thing is being available. I feel like all of my mentors, I'm only now realizing how busy we are on faculty and how many hats we wear and how many emails we get. And it's just, the more I get into my role as faculty, the more I realize how generous my mentors were with their time, and were always available to listen. And they knew when to listen, they knew when to give advice. They seemed to just push at the right times and listen at the right times. And I'm just learning how to balance that. But I think being available and looking for opportunities for your mentees, and just opening the doors for them, as you said, is what really makes a great mentor.
Yeah. Well, if you figure out how to do the balance, please share, because all of us are probably not very good at this balancing aspect. We tell fellows all the time, oh, it looks pretty easy to being a staff consultant, or what we call the consultant. And oh my gosh, as you said, there's just a lot of extra things which we all want to do. For us in academics, and most of us are in academics and doing research institutions, publication and grants are the currency of success. And I know it's important to do that. And you got involved pretty quickly with clinical trial work. And maybe how did you get interested? You went to the mechanism aspect and you are now involved with clinical trials. And how did this all come about?
I think, again, just having the opportunities to be involved. So when I was a fellow in the later part of my fellowship, after I had accepted the position here and went, oop, I better get some trial experience, Dr. Riedel had me reviewing some protocols with him that were going up for review at the IRB. And that was really helpful to see how our protocols put together, what is the design like, what questions come up, what concerns come up at the IRB. So this was something that was really helpful. I think then coming here, we had some education around clinical trials and being a PI, and that was really helpful to get started as well. But I think nothing prepares you to just do it. And the more I've been involved in studies, even there's a lot I'm learning from industry-sponsored studies too. And just the more I do it, the more I feel more comfortable in that space. And now that we have SARC 44 open and another study that I'm leading through CTEP that's looking at peposertib plus low-dose liposomal doxorubicin and leiomyosarcoma. With these two studies open, as the study chair, I now feel like I'm really starting to understand what is a trial, what things we need to look out for. So it's been great to see all of that finally out there in the world, open and enrolling patients.
Well, you think about it today, 3 years later, you'll have it up and running, and that takes time. Okay,
That's
as you
true.
know, and we're not here to compare SARC versus CTEP, but it's great that you are a national PI on national studies. And that's where we all want all of our colleagues to be. With that in mind, maybe you can share with our group, what is SARC 44, since we've said it several times in our discussion, and because every time I hear about it, the flashing of excitement starts coming in there. But maybe we'll just start. What is SARC 44, first of all, and then what is the trial trying to study?
Yes, great. So SARC 44 is a phase 2 trial. It's a single-arm study investigating bezuclastinib and sunitinib, both TKIs, tyrosine kinase inhibitors, in patients with GIST that have already been exposed to sunitinib, which is usually our second-line drug. And the rationale for this study is essentially that, while imatinib and other tyrosine kinase inhibitors have really revolutionized how we take care of patients with GIST, we know that they ultimately develop resistance to treatment, and that those resistance patterns that we're seeing are raising questions in the field about whether, we need to tailor our treatments and select TKIs that specifically match the resistance patterns, or if maybe a broad approach that covers more resistance mutations is the best approach. Not that these two need to be compared. Maybe they both have a role. At any rate, the PEAK study is ongoing now. That's a phase 3 trial that's looking at the combination of bezuclastinib and sunitinib in the second line. But when this combination was first being tested, the first in human study included patients that had had multiple prior therapies. And we could see that the activity of the combination was even good for patients who had previously progressed on sunitinib. So that suggests there could be a role for this combination in later lines. But it was still a very, early study. Few patients were included. So the goal of SARC 44 is to look at the combination in those patients that have already had sunitinib and likely many other therapies too, based on the population that will enroll. So I expect that this will be a more heavily treated group of patients, and we hope to see that there's good activity even in that group of patients. The other key thing with SARC-44 that I think is a real star is the correlative work that's going to go along with it. So overall, we're going to enroll 40 patients, but there's going to be a subset of 20 patients that will have a series of PET scans. And then those PET scans are going to inform a selection of a tumor to biopsy. And with that information and with that biopsy sample, we can learn so much about resistance patterns and GIST, resistance mechanisms, or response, why certain patients do better than others. And this will give us a lot of information. I think that's even going to extend beyond SARC-44 and this combination, but may inform the field more broadly about resistance patterns and GIST and what the next line of therapy may look like.
And that's actually a very nice summary, because this is a key part to say is it okay to add this medicine to a drug that you've already failed or progressed on in the past? And if we can resalvage it, that would be great. You're highlighting an important aspect of a lot of our newer SARC studies, and that newer SARC studies is trying to answer biology questions. Not does it work or not, but why did it work and why it didn't? And this almost gets back to your pipetting days, where this is all heavily scienced out, but you had a couple mentors that helped get this over the finish line, as you say. Maybe you can share a little bit about that, because this study, was brought to SARC with a lot of good input, but it didn't have to come to SARC in that, but maybe you can share.
Yeah, so Dr. Andy Wagner has been a really key mentor on this study. From the earliest days when I first joined Dana Farber, he had this on my radar, and he looped me in very early and talked about the data that he was seeing with the study he had just published, the first in human study, and about why we would be interested in looking in the population that SARC-44 is enrolling. And then he basically mentored me the whole way through. We've designed the study together. We've talked through the correlatives together, partnered with Dr. Jonathan Fletcher, who's also been a great mentor. Yes, Andy deserves extra special mention. He's been a really wonderful mentor throughout my time here.
No, actually, exactly correct. And if you put a little bug in the ear, Megan and I have been trying to get him to do a podcast, and he said, "Oh, Dr. Haddox, I told him that we're going to do one." He says, "Oh my gosh, I better hurry up and get on there too." So you beat him to a podcast. So, yeah. But it's important, and you're leveraging the strengths of Dana Farber. You got Jonathan Fletcher, who is the scientist, helping that mechanism thing. You got Andy Wagner, who's been through the ringer many times and trying to get these studies over the line, and it's taken some time. And we're very fortunate that Aaron, part of the SARC team here, has been helping shepherd you, trying to work through the first protocol, get activation. You're smiling and shaking your head because, oh my gosh, this takes a little bit of time and effort. And oh, by the way, we got to make some adjustments because queries are coming in. It's like we thought we knew everything, but it's a constant evolution process. So we look forward to, finishing out, getting all the sites up and going and completing this study, and hopefully it's positive. But even if it's a negative study, it's positive that we'll get some good science out of it as well.
Yes, definitely. And as you mentioned, it takes a village to get a clinical trial off the ground. And I'm so grateful for all of SARC's assistance and support, and really, as you said, shepherding me through this. You all have been wonderful. And I look forward to, I know, one of the benefits of working with SARC is also for that extra support and mentorship and community. And I look forward to discussing the study as we get up and running, as we start to get data, because I know that, the individuals at SARC are going to be key in giving me input and feedback on how things are going and interpreting all the data that we're getting in.
We're excited. So we're excited to get this study up and running, and hopefully we can accrue fast, and we'll get some answers. And you're right. We'll all learn together on this. And I think we're almost at a point where we're kind of saying we're all colleagues, so we're all supporting each other in this effort. So this is really a fun time. As we kind of get to the end, the close here, looking ahead, maybe 5 years from now, when you and I are sitting here having our 5-year reunion in SARC Talk and podcasts with each other, what new things about sarcoma or what new things about your career should we be talking about? Kind of what's your vision in the next 5 years? So when we come back, we can say, yep, that's exactly what she hit, or she passed that going 99 miles an hour.
Yes, I hope in 5 years we have data back on SARC 44. But no, I think to be aspirational, I hope we're talking about SARC trial 144, perhaps a mechanism, a drug that we haven't even come across that mechanism yet. Hopefully there's that much more progress and advancement. And I hope to be here to lead those studies and be involved in biomarker development. And that's my goal, is to help improve the number of treatments, the tolerability of the treatments, and select the patients that are most likely to benefit to get those treatments. And if I can achieve all of that in 5 years or 30 years, more likely, then it'll be a success.
Well, it's going to be a success because what you're doing today will help our patients in the future and help us avoid unnecessary or appropriately necessary treatment, decisions in the future. So we look forward to your success, and we'll be following you because you're one of us. You've always been one of us, but this is even more exciting. So this is good. Dr. Haddox, any additional final thoughts from your end?
I would just say I'm looking forward to the next steps, the next years in my career, and look forward to further partnership and moving things forward for our patients.
Yeah, perfect. I was hoping you'd say you're looking forward to better IT support, but I didn't hear that coming through right now. And that's an inside thing because we've been struggling a little bit with IT. So hopefully this comes out. People can see us. Maybe not, but at least they can hear us. And it's really great, Dr. Haddox, that you got SARC 44 under your belt, and you've shared a lot about how you got here. And many of us came here just by hook or crook as staff, not in medical school. So you've had a very nice journey. So we look forward to hearing more about it.
Thank you.
Thank you all for listening, and we'll be back for another SARC Talk episode when we interview interesting people who are connected to sarcoma, and where we can all share and spread the knowledge of sarcoma. And we look forward to our future podcast together. So thank you.
Thank you for listening. Be sure to subscribe so you never miss an episode of SARC Talk. To find out more about SARC, please visit our website at sarctrials.org. To suggest a topic or for questions, please email us at sarctalk@sarctrials.org. The SARC Talk series is made possible due to funding from Boehringer Ingelheim. Together, we can find a cure for all forms of sarcoma.